All right, so we talked about the non-cyclic pathway, okay, which is the more widely used. Okay, but there are times when a photosynthetic organism may switch to a cyclic pathway. Okay, so we need to talk about that. In the cyclic pathway, only photosystem 1 is used, not photosystem 2, then photosystem 1. Only photosystem 1. So photosystem 1 will absorb a photon and emits electrons. The electrons ejected from photosystem 1 enter electron transfer chain. Okay, hydrogen gradient forms across the thylakoid membrane to drive ATP formation. And then the electrons that reach the end of the electron transport chain, instead of being accepted by NADP, they get returned to photosystem 1, so the electrons are recycled. Okay, all right. And so you don't have NADPH forming. You don't have the splitting of water because electrons don't need to be replaced. So you don't have oxygen forming. In cyclic pathway, you only have ATP being produced. Okay, all right. So you only have ATP being produced, no NADPH, no oxygen. Okay? Why? What's the purpose of this? Okay, if you don't have NADPH being produced. Well, this Calvin cycle uses a lot more NADPH than ATP. Okay? All right. And so, let's take that back. The Calvin cycle uses a lot more ATP than NADPH. Okay? All right. And so you can have a buildup of NADPH, okay, in the plant cell, all right, but a depletion of ATP because the Calvin cycle uses more ATP than NADPH. Okay, and so to try to catch up that, to replenish the ATP, they just undergo cyclic electron flow. All right, and that way more ATP is being produced, but not more NADPH, which they don't need. Okay. All right, so you need to know the difference between cyclic and non-cyclic electron flow. All right, and the reason why plants may go from non-cyclic to cyclic. Okay, and the main reason is because the Calvin cycle uses a lot more ATP than NADPH. Okay, so here you go. Here is the roles of non-cyclic pathway and cyclic pathway. Okay, so important information to remember from photosynthesis. Okay, but once NADPH is produced, then they go to the light independent reactions called the Calvin cycle, Calvin-Benson cycle, whatever. And here is where you are building the food. All right, here's where the photosynthetic organism is building the sugars that it's going to use for energy later. And it happens in the stroma of the chloroplast. It's light independent reaction, so it's not powered by light. But the driving force is the ATP and NADPH that was formed in the light dependent reactions. Okay, so where does it get the carbon to make these sugars? It gets the carbon from CO2. So the photosynthetic organisms will take up CO2 from the environment, and they will use the CO2 in the light independent reactions to form the sugars. Okay, and that's you may also say it's called the carbon fixation cycle, because through that process of using carbon atoms from carbon dioxide to make sugars, it's called carbon fixation. Won't really get into the whole lot of the Calvin business cycle. But here's some of the highlights. You have Rubisco. Okay, Rubisco is a very important enzyme. It's one of the most abundant enzymes on Earth. Okay, and so the first step of the cycle is that Rubisco attaches CO2 to a 5-carbon molecule called RUBP. All right, and then eventually this 6-carbon molecule will split, and then that's when you get sort of the building of the sugar molecule. Okay, all right, but again, it's a cycle, and so if you're using this RUBP in the first step, you got to remake the RUBP, and you do see a recreation of RUBP so that it can cycle through again. All right. So the big things here, don't necessarily worry about all this. Know that ATP is used, energy from ATP is used, the electrons from NADPH are used, and hydrogen ions. Okay. All right. And then the product of the Calvin business, the running of the Calvin business cycle, are the sugars that the photosynthetic organism will use for energy, for food. Okay. All right. So it occurs in the stroma. You got to have an input of carbon dioxide. Okay, because it needs that to get the carbon to build the sugars. Okay, the first step is carbon dioxide is added to RUBP by that enzyme Rubisco. You have a regeneration of RUBP so it can run again. Uses ATP for energy or ATP is used to provide energy to fuel the cycle. And then it requires the electrons from NADPH to run as well. Okay. All right. So that's basically the overview of photosynthesis. Okay. The major steps. All right. So now we're going to kind of finish talking about how some plants do photosynthesis differently. Okay. And so to talk about that, we need to talk about the structure of leaves. Okay, so stomata are tiny closable gaps on the surfaces of leaves and stems on a plant. And when those openings are opened, when the stomata are open, it allows carbon dioxide to come into the organism and it allows oxygen to leave. Okay, it also can lead to water loss, which the plant doesn't want. All right. And so when we have those hot, dry days, okay, those stomata will close to conserve water. But when they're closed to conserve water, that also means carbon dioxide can't come in and oxygen can't leave. And so you're going to see a slowing of the light independent reactions. And so less food is made. All right. So here you see stomata on the surface of a leaf. You have different types of leaf tissue, that you'll talk more about in biology 102. Okay. But within these tissues, you'll have your, within these tissues, especially within the palisade mesophyll, you'll have a lot of chloroplasts, and that's where most of the photosynthesis is happening. And then the spongy mesophyll, you have these kind of open spaces that you see. All right, that's where that gas is stored, carbon dioxide is stored until it's needed for photosynthesis. All right, well, the issue with closing the stomata for plants is that they can develop photorespiration. Okay, and so photorespiration occurs in, it's a big problem for C3 plants, and C3 plants are your most common type of photosynthetic plants. Okay, so their stoma will close on hot, dry days. So that means no carbon dioxide is coming in. Okay, and if when no carbon dioxide is coming in, that Calvin cycle is still wanting to run. Okay, and so what will happen is that rubisco, instead of attaching to carbon dioxide, it will attach to oxygen, which also can't leave the plant because the stoma are closed. All right. Well, oxygen isn't good for running the light independent reactions. Okay. All right. And so you have a lot less food that's being made. All right. And so, and then you also have a lot less ATP being produced as well. So photorespiration is a big problem for C3 plants on those hot, dry days. Okay. So we said photorespiration is inefficient because when oxygen levels rise inside the plants because it can't leave through the closed stomata, the rubisco will attach oxygen to RUBP. Okay. And then that has to be dealt with by the plant, and it's a lot more inefficient. And while the plant deals with that, or during the dealing of the oxygen binding to RUBP, it has to use ATP. Okay, and so that's why that leads to the inefficiency, because it's losing energy that it doesn't have to lose on a normal basis. Okay, and so it's inefficient. All right, so how do C3 plants compensate for photorespiration? Well, their method of compensating for photorespiration is to make a lot of rubisco. And if you make a lot of rubisco, the more rubisco you make, the more chances that they will bind to even small amounts of carbon dioxide. So hopefully you can still have some Calvin-Benson cycle running to produce food. Well, you have other plants that have adapted to this problem of photorespiration. Okay, they have evolved to minimize photorespiration, and you have C4 plants and CAM plants. Okay, now you may wonder, well, why is it called C3? Well, if we go back to here during the production or during the running of the Calvin-Benson cycle, you have these 3 carbon molecules being produced, okay, called P-gal. They're just 3 carbon molecules. And so that's why they're called C3 plants. Okay. All right. So these C4 and CAN plants will still close their stomata on hot, dry days. But even though their stomata are closed on those hot, dry days, the sugar production remains high, and that is because of their adaptations. Okay, so C4 plants, they don't have as much photorespiration because they produce an additional enzyme. So they produce PEP carboxylase in addition to rubisco, and PEP carboxylase has a higher affinity for carbon dioxide than rubisco. Okay, all right, and so the PEP carboxylase will bind to carbon dioxide, produce a 4-carbon molecule, okay, which can then be used to enter it into the Calvin-Benson cycle. Okay, so that's why they're called C4 plants, because this enzyme PEP carboxylase is involved in the production of a 4-carbon molecule. So common C4 plants: sugarcane, corn. Okay, plants that we know, but they have adapted to this issue of photorespiration. Okay, so again, PEP carboxylase it's just an additional step, right, to produce these 4-carbon molecules, and then will be entered into, or basically carbon dioxide can then be taken from to get into a Calvin cycle. And then finally, you have CAM plants. Okay, now CAM plants are really adapted to dry conditions. Okay, so a lot of desert plants are CAM plants, very arid conditions. Okay, now what they do is they will open their stomata at night and close during the day, during the hottest parts of the day. Well, that presents a problem because you open your stomata at night, cooler, okay, so less water loss. You can get carbon dioxide, but when do the light-dependent reactions occur? They have to occur during the day because the sun fuels the light-dependent reactions. All right, so there's the conundrum. All right, so how do they deal with that conundrum? Well, they make an organic molecule. Okay, they make that at night, and while the stomata are open, and then this organic acid is used as a carbon source during the day when the light-dependent reactions are going on. Okay, so at night you have these organic acids being made, and they're stored by the CAM plants until the daytime when the light-dependent reactions can occur to make ATP and NADPH to fuel the Calvin cycle. Okay, so this just kind of shows you the comparison between C3 plants, which are the most common, C4 plant, and CAM plants, okay, which have adapted to this problem of photorespiration. All right, so need to know, compare the overall reaction of photosynthesis to aerobic respiration. And what you should see is that depending on which way the arrow is pointed, is you have either photosynthesis or aerobic respiration. Where do they occur? Okay, where in a cell does aerobic respiration occur, photosynthesis occur? What are the major steps of photosynthesis? Being able to summarize the major events in each step. Okay, what are the differences between cyclic and non-cyclic electron flow? Okay, what are the major products between the two? Why would a plant undergo cyclic versus non-cyclic electron flow? And then be able to compare and contrast the light-dependent reactions and electron phosphorylation in aerobic respiration. Specifically, what I'm talking about is those electron transfer chains. How are they the same between the light-dependent reactions and the electron phosphorylation, and how are they different? Okay, what is photorespiration? And then how have those C4 plants and CAM plants adapted to this problem?